Thanks everyone for joining today. I wanted to just sort of say good morning to everyone. I'm Erica Lotz from Stantec, and I'd like to welcome you all. So in non-COVID times, I sit in the Burlington, Massachusetts office, and I'm part of Stantec's water leadership team for the Northeast in the US. And so also with us today is Katie Chamberlain, who also works with me in the Burlington, Massachusetts office. And Katie is Stantec's water treatment sector lead for the Northeast. And a few months ago, Katie and I were sort of chatting about some of the challenges that face water suppliers, and what could we kind of put together to help along these lines. And so we were discussing harmful algae blooms as a subject that is a challenge for a lot of surface water suppliers. So we reached out to Stantec's research group and pulled in Nicole McClellan, who will be presenting today with us. And she'll be kind of talking about some of the research and some of the evaluations that she has done regarding harmful algae blooms. So it sounds like, as we were reaching out to folks about this webinar, it seems like it's kind of well timed. So I think there's some communities out there that are being challenged with algae blooms, even right at this point in time. So welcome everybody. Just as sort of things to keep track of, we're going to ask if folks could turn off your cameras during the session, and our presenter Nicole will have hers on. That just keeps down some of the distractions that you get, right, staring at other people and what are they doing. So and we're also going to ask that folks hold their questions to the end. If you would like, you could type your question into the chat feature on Zoom, and or you could raise your hand using the raise your hand feature on Zoom. And then at the end, I'll kind of moderate and call on folks to ask their questions. So I appreciate everyone's time to sort of listen to us today. And so presenting today on algae blooms will be Nicole McClellan. Nicole is a water treatment process specialist with over 13 years of experience related to the management of contaminants of concern and treatment optimization. She applies her background knowledge of biological systems and civil engineering to provide solutions to unique and complex challenges such as harmful algae blooms. Nicole has developed site-specific, user-friendly models to estimate the removal of contaminants of concern, such as cyanotoxins, and to inform management decisions to ensure regulatory compliance. And with that, I will turn it over to Nicole.
Great, thanks, Erica. Thanks for having me today. It's really my pleasure to share the information that we have based on our experience of providing some solutions related to algae blooms to some of our clients. I'm based in Waterloo, Ontario, Canada, but we've helped some utilities across the US, and we're seeing that that's becoming more common now as we're having a warming climate and more pollution in various areas. And so so we're trying to be proactive. So I think this is a good way to do that. This will be a good reminder for all of us, a good check-in on how prepared are you if there was an algae bloom in your water supply. So the focus for this presentation will be mainly on the management of the cyanotoxins within the water treatment plant, as well as the cyanobacterial cells themselves, and some consideration for algae cells in general and the taste and odor compounds or contributions that they can make. And I'll talk a little bit about the management of blooms at the source, but most of that material I've left for our environmental studies group, who can often help us with those remediation efforts as well. So Stantec, we always like to start off with a safety moment. And now that we're getting into some colder weather for many of us, I think it's a good reminder that we try to stay healthy and fit, especially for those of us who are still kind of restricted with where we can go, thinking about what you can do that's safe with your family, exploring green spaces, being outside as much as possible, thinking about how you're going to stay active through the winter. We're really lucky here. My son's been able to go rock climbing indoors, so I hope to keep that up. Hopefully they can stay open, fingers crossed here. It's a good reminder there. So I think it's good to ask ourselves, how prepared are you at your utility if there was a bloom to form, a harmful algae bloom, which I'll describe more in a minute, in your source water supply. Often this can happen overnight. Winds can stir up algae from the sediment, push them into these shallow bays or shores where the water is warm, and there may be higher nutrients, they're closer to the sediment there. And you've got lots of days of sunlight and low wind. And now you have all of a sudden this toxic algae bloom that may be coming into your plant, which probably many of us are familiar with what happened in the late summer of 2014 in Toledo, Ohio. At the water intake, nearly 500,000 consumers were told they could not drink, cook, or bathe with their water. And that's often what does happen with these types of events, is that we can't boil away the toxins, and they can also be an irritant for skin. Like they can have dermatotoxins in the blooms. So we really can't use that water for anything when we're concerned that toxins might be present. And so you need to think about what might be your emergency response. Can you switch to emergency supply? Are you able to provide water trucks or distribute bottled water? Making sure you have all those contingencies in place, because these things can happen very quickly, especially if you don't have a very detailed monitoring plan in place for your water supply. Your lake or a river even could happen in a river as well. So the cause, of course, for Toledo was a harmful algae bloom or algal bloom. We say algal because it's associated with algae, but it's actually cyanobacteria that are producing these toxins. And it was this, this microbe itself, Microcystis aeruginosa, at this event, producing the toxin microcystins, which many of you may be monitoring if you have experienced blooms at your source before. So, so just something to think about as we go through the presentation, maybe making a checklist to follow up on and seeing where we can help as well. So some of the problems that can come from algae blooms, the reason why this is so important, many reasons actually. It's a very complex issue. Often when we're dealing with other contaminants of concern, I know PFAS has become really top of mind. Aluminum regulations are getting more strict in Canada, these kinds of things. But when we're dealing with chemistry, we can often look at the speciation and predict what might happen. But in this case, we're dealing with a living organism, something that can multiply kind of like E. coli. So it makes it a lot more complex. And so hopefully throughout this seminar, we'll be able to weed through some of that complexity and make it simpler for you to understand some of the information that's coming down. So some of these issues, they can produce toxins, of course, that cause illness, not just to humans, but also livestock. And, certainly you don't want your dog or children wading in these waters, can make them sick as well. Can lead to taste and odor issues, and not just the harmful algae blooms, but any algae bloom can result in taste and odor issues. So if you are having taste and odor complaints, you may want to see if it may be caused by algae. It can interfere with the actual performance of your conventional processes, specifically, floc formation, filtration, chlorination, or increasing the DBP precursors. And so if you're seeing those on the rise, you might want to check that it's not algae. And of course, there's, more watershed type impacts, like the pollution of our source waters and the impacts on our economy and tourism. Nobody wants their water to look like that. So today we're going to really weed out what is the difference between an algae and a cyanobacteria, and who's making these toxins and what are the other concerns. We'll look at what are some of the causes that might help you build a better or more robust monitoring plan for your utility. And then I'm going to touch on a little bit of how we might recommend initially managing HABs in the source. And then my focus is really on the performance of your treatment processes and how well they might be able to remove these toxins. It's possible that your existing treatment train is well equipped to remove many of these toxins, or could just require some tweaking. Or it's possible that additional treatment processes could really build up your robustness for a harmful algae bloom. So we have algae, and then we have cyanobacteria, and they are different. And you've probably heard the term blue-green algae, and I think that's what makes this so complex. And that's what we used to call cyanobacteria. Some references I saw, EPA still sometimes refers to cyanobacteria as blue-green algae. So that's what probably makes it very complex, because they're not a plant, they're not an algae species. So we have these algae species, they're plants, they're typically multicellular, they're more macromolecular, they're larger when you see them in the water body. They do produce energy by photosynthesis using chlorophyll, of course, being a plant, and they're naturally found in surface waters. So they're not necessarily an indication that your water body is unhealthy if they're just existing in low numbers. But they can multiply, and they can form non-toxic algae blooms. So it would be a non-harmful algae bloom. So you may see what you think is a harmful algae bloom, but it's actually just being produced by algae or an overgrowth of algae cells, and not producing any harmful toxins at all, although it could contribute to taste and odor events. So still a concern for utilities, but from the human health perspective, less of a concern. And then we have these other species in our water bodies, also naturally occurring, and they're cyanobacteria. So there's a bacterial species that acts like a plant. Being bacterial, they're typically unicellular, although they do like to form these sort of chain-like or cluster formations of multiple cells close to each other. They also produce energy by photosynthesis, so they do have chlorophyll, and that's where this is sort of a bacteria pretending to be a plant. And often we will find cyanobacteria, again, naturally in water supplies in low numbers, and they're not causing any problems. They may be producing toxins, but they're in such low concentrations that we wouldn't detect them, and it wouldn't be a concern for human health. But they can also form blooms under the right conditions and produce harmful toxins known as cyanotoxins, microcystins being the most common that you've probably heard of. And then these are the species that can actually multiply and form what we call harmful algae or algal blooms, these HABs. So that's what we're really concerned about from the human health perspective. And of course, these can also, they're made up of organic matter, so they can contribute to those taste and odor events as well, and impacts on treatment performance. So a little reminder of our photosynthesis pathway here. These species both will thrive with lots of sunlight, and with pH changes, having lots of carbon dioxide present can really push their growth into bloom formation. And then they're producing oxygen, but then of course they'll respire in the absence of sunlight and produce CO2 and take up oxygen, which can happen, as the bloom is decaying, and we have bacteria that will thrive on that matter and consume the oxygen, which can result in fish kills. So that's sort of, moving into more of the eutrophic condition in a water body at the end of the bloom. So there's some key cyanotoxins of concern, not just microcystins, although it's the most commonly detected, the most commonly regulated worldwide. And there's over 100 variants of microcystins now detected. I think just a few years ago, we only knew of maybe 80. So we're always learning about more of those. And I think there's at least 10 under the candidate contaminant rule, list number 4. And we can see that there's many species that produce these toxins. Some species of cyanobacteria produce more than one toxin. And they're not, on the other hand, they're not always producing toxins. So it's another level of complexity that when you detect one of these species of cyanobacteria in your water supply, it doesn't necessarily mean that you're going to have a high level of cyanotoxins present. So you'll want to be monitoring for the cells. That's a good approach to see, what species you have to know what type of toxins maybe you'll find in your water. But you also want to be monitoring for the toxins themselves. And there's some molecular approaches. There's qPCR assays that are accredited methods where you can look for the toxins, and also ELISA dipstick, not really dipstick, but test strip tests that you can do in the field to look for the toxins as well. So again, kind of taking a multivariate approach to monitoring for algae blooms. And the main concerns with these toxins are that they can affect many organs in the human body. Mainly microcystins and Cylindrospermopsin are hepatotoxins, meaning they're cytotoxic to the liver. And we know this because there's a large body of research out of China where there are persistent algae blooms in their drinking water supplies. And we see these effects from low dose chronic exposures over time, that people in these communities will develop these types of illnesses and have these effects. And also from, other mammal lab studies, we understand the molecular mechanism of these toxins. So we really want to avoid even low concentrations over a long period of time. And that's one reason probably for the US EPA's 10-day health advisory is, you don't want people being exposed to these low concentrations for more than 10 days at a time. And then there's other toxins that are anatoxin A and saxitoxin. Some of these are also on the CCL4, and those are neurotoxins. And we know this again from some human exposures. There's a really good case study of an unfortunate surfer in Brazil who took a gulp while he was surfing and had some health effects, survived. But these kind of exposures give us some insight on what the health effects are from these toxins. So pretty serious concerns, especially for the young, who are, consuming more water per body mass. So we have different regulations for young children under 6 than we do for adults. Well, also on the slide, I'll just mention that we can see the molecule microcystins here, and it's actually a very large molecule. And all of the variants here, you can see MCLR is like the most common, and that's named because of these amino acids that are in place on this particular molecule. And knowing that it's a large molecule helps us understand how effective something like PAC or GAC might work. We need to have fairly large pores in that media for it to absorb toxins like microcystins and Cylindrospermopsin that are pretty large molecules relative to other toxins that we might be worried about in our water. So I wanted to help you guys understand, what's the persistence of these toxins in a bloom, and another level of complexity for algae blooms. So, this is our well-known Lake Erie, for example, has many well-known algae blooms. And so within that bloom, we may have several different species, for example, here, Microcystis colonies or oscillatoria, that are capable of producing cyanotoxins. And those toxins at the beginning of the bloom are mostly going to be contained within the cells themselves, within the cell wall. So we call those intracellular or cell-bound toxins. And then we're pretty sure that the toxins are not actively excreted by the cyanobacteria themselves, but they are released as the bloom breaks down. So there may be some stressors to these cell walls, like a preoxidant, for example, or just natural die-off or predation, or physical rupturing, that kind of thing, from maybe sonication or chemical attack, that could release those intracellular toxins to become extracellular. So coming into your water treatment plant, you may have both states of the toxin, some that may be bound within the cells and some that is actually dissolved. These molecules readily dissolve typically in the water column. And so we have these two challenges coming into our water treatment plant that we have to manage. And it's probably in your best interest to deal with intracellular toxins while the cells are intact, to actually remove the cells themselves along with the intracellular toxin, and then deal with the extracellular remaining fraction. So we want to prevent any rupturing of those cells as they're coming into the plant, try to get those cells out through, DAF or conventional treatment or membranes, and then deal with the extracellular fraction, typically with oxidation or adsorption processes. Typically, these toxins can persist in the water column for, it's been reported, up to a couple of weeks after the bloom dissipates. So we definitely want to be keeping an eye on the state of the bloom and how it's dissipating, as well as measuring for the actual cyanotoxins themselves. So we may not be out of the clear very quickly. You may be relying on an emergency water supply for an extended period of time before you can either upgrade your treatment processes to manage the toxins or flush your system and while you're relying on this emergency supply. So what are some of the things you can look out for in your source water that may be contributing to algae blooms? This is a fairly simplified concern. Typically, I would say to a utility, just walk out and walk around your water supplies and look at what some of the risks might be, and then start to dig into the nitty-gritty of the data that you have. Certainly, excess nutrients will be providing a food source for cell growth. So if you have supplies that are heavily impacted by agricultural runoff, with fertilizers and animal waste, those nitrogens and phosphorus compounds are going to be great food sources for these algae blooms. You may have chemical discharges and waste from industry or septic discharges. It's very common in cottage countries to have these algae blooms on small lakes where there's uncontrolled septic, or even stormwater can have higher concentrations of phosphorus and nitrogen at times that can contribute to a bloom occurring in a short period of time. Of course, as these guys are photosynthetic, sunlight and temperature, extended periods of high sun and warm temperatures with low winds are really going to be favorable for a bloom to form. And we don't want these conditions that lead to stratification within our reservoir. We don't want low dissolved oxygen or poor mixing in our reservoir that can really lead to conditions for bloom formation, particularly on the surface of the water supply. One of the contingencies that you might look to is having, depending on the depth of your reservoir or your lake or your source itself, is thinking about having different intake levels. In the event that you have a surface bloom, then you can pull from your lower intake or vice versa if the bloom is forming in the subsurface. And different cyanobacterial species will propagate at different levels in the water column. So we know Planktothrix, for example, it likes to form in this, hang out rather, in the sediment. And then if you have a wind event, it can stir them back up into the water column. Other cyanobacterial cells have little air pockets that allow them to control their buoyancy. And so during the day, they'll move up to the surface to perform photosynthesis, and then they'll move back down to the sediment when the sunlight isn't there and just save some energy and multiply down there. So it's good if you're doing physical, visual monitoring of your water supply, which we do recommend, that you're doing that at different times of day so you're not missing potential bloom forming as well. So we can see, how thermal stratification may occur, particularly in the summer in your water supply. And so we don't want to have, a large degree of temperature change. So you may be interested in installing some remote sensors for temperature throughout the, say every foot depth within your reservoir to monitor the temperature to see, is your reservoir experiencing stratification at particular times of year, and is that contributing to or correlated with algae bloom events? Then you may want to look at, a method to address that. And so when we have these conditions with stratification, we can get low oxygen concentrations in the subsurface, which can lead to more nutrients becoming dissolved in the water column that act as a food source for the algae, particularly in the summer when the water is warm at the top of the water surface or in the upland. And we also have some concerns about, how the algae might, I was talking about the intakes at different depths. So if your intake is located where the algae are accumulating in the water column, colleagues of mine at the University of Toronto had looked at, what if you even have low concentrations of algae cells, or cyanobacterial cells coming into your water treatment plant. So they looked at plants where they didn't necessarily have visual blooms, but they did have the presence of cyanobacteria under these favorable conditions coming in. And they found that those cells were accumulating within their treatment plant. So they were accumulating in the clarifier surface up to 2,500 cells per mil. Those cells were accumulating in the sludge. And if you're not practicing frequent sludge cleaning, then you could have the cells sort of decaying here and releasing their intracellular toxins to become extracellular and increasing that concentration of toxins in the water column. And then they've also seen the cells accumulating within the filter surface and in the filter media itself. So even if you have, you haven't seen any visual blooms in your water supply, you may want to, look at occasionally, maybe quarterly or monthly throughout the warm bloom risk season, looking at getting some cell counts done for algae and monitoring those toxins routinely as well. So one of the questions we get asked a lot, just make sure I didn't miss a slide here, is RHABs on the rise? And at least for Lake Erie and in Canada, we have seen that this is becoming more of a concern. We can see this is the NOAA, their forecast for the bloom severity. And we can see since 2002 that the bloom risk has been, higher than past years, past decades, and, more into the significant category or likely to experience a bloom. And we do believe that this is related to gradually warming temperatures, as well as more nutrient input, and also pH changes seem to be contributing to algae blooms. We have less acid rain here. We've cut out coal in Ontario, and so we know that's also happening in Atlantic Canada, that there's pH changes happening in the water supplies, the surface water supplies specifically, and that may be contributing to conditions more favorable to certain species of algae. So even if you haven't experienced algae blooms, we definitely want to be keeping an eye on that. Or if you think you're in too cold of a climate, certainly even under ice, we have observed algae growing, in Sudbury. I was talking to a colleague a couple weeks ago who studied algae under the ice in Antarctica, because those conditions have a low change in the temperature. So it's a nice, consistent climate for bacterial species to live in. So we want to just be keeping an eye again on that. And as I mentioned earlier, I won't get into too much detail here, but we know in these cottage countries, we see where people don't have municipal sewage treatment or management, that there may be blooms associated with those nutrient inputs from uncontrolled or poorly managed septic systems. So we want to, understand that that's a contributor, and then look at how we can better manage that. I'm getting a little close to my time, so I'm going to just speed up a little bit here. But there are some really good resources out there that I'm going to mention throughout the presentation, and there's some really good information from this Clean Water Action Fund that tells us a little bit how you can assess your source water, your drinking water system, and help inform the public, and again take more of a multivariate approach to this very complex issue. So for example, at your source, you'll want to be thinking, are we affected by drought? That's definitely been contributing to more frequent or concerning algae blooms in California and Texas in recent years. Are your customers experiencing odors? Are you having more odor complaints? Is your reservoir stratified? These kinds of things that we've talked about. And we're going to talk about how to better manage or be prepared for an algae bloom through the drinking water treatment system. So we want to look at, what's your local legislation? How can you develop a, excuse me, an effective monitoring and mitigation plan? And then what's your action plan? Your response to communicate to the public through social media or other avenues, and how you're going to respond with respect to actions at your plant as well. So looking at the drinking water regulations, if you want to call them that, in the US, we don't have any federal regulations for cyanobacteria or their toxins at this time. You may expect that to come down the pipe potentially. This is definitely one to keep an eye on, that we're going to see some changes as there's many of these contaminants on the CCL lists that are being monitored. At this time, you do have your 10-day health advisories in place, as I mentioned, lower targets for, excuse me, children under 6 and people over 6 and adults, having those higher concentrations. I did find some more stringent or specific values in Vermont. They also have a value in place, a health advisory for anatoxin A. And so if you are detecting anatoxin A, you could look, even if you're not in Vermont, look to what this guidance value is. And Ohio, since they are on Lake Erie and they have had some persistent bloom issues and toxins passing through their water treatment plants, they have some action levels that they've established. In Canada, we have one value, which is for total microcystins. So that's for all the microcystins variants that we know of. And it also includes the intracellular and extracellular. So when that sample is being processed, all of the cells are to be lysed or ruptured before the analysis is done. And that value is 1.5 micrograms per liter. And then the World Health Organization has released provisional values this many years ago for just MCLR, just this one variant, the most common, of 1 microgram per liter. So you can kind of see what's going on in the rest of the world, and these values are kind of in line with those for sure. You can see that, this one for younger children is a little more progressive for microcystins than the Canadian one that is typically adopted by most of our provinces as a regulatory standard. There's some new regs coming out for recreational water that I didn't include here, but we can definitely talk about that later. The EPA has released some really good visuals that you can use for, these are a little older now, but certainly still relevant to the current health advisories that were established, that you could use for your staff or for your media announcements. And there's also many templates available for you to develop your public notices for each of these action levels. If it is only for vulnerable populations, or if you do actually have to issue that do not use and for how long, and explaining why, there's some good templates and narratives that are available, and we can help you develop those as well. So for your monitoring plan, if you're thinking about beefing that up, you can adjust it seasonally. We do have some utilities that have blooms, in March and October, November, even in Canada. So we don't necessarily want to taper it off too much by season, but certainly, in the very colder months, we can maybe conduct monthly or bi-weekly sampling versus weekly or daily. So we want to be looking at how can we identify conditions favorable for the algae growth itself and the cyanotoxin blooms. So we can have these remote sensors for the toxin pigments, or the, sorry, this should say cyanobacterial pigments, not toxin pigments. So we can monitor those by looking for the chlorophyll or phycocyanin, which is a pigment that cyanobacteria produce. We can look at, is your utility, your source water supply experiencing stratification, and these other water quality conditions that might be related to blooms, like the phosphorus concentrations, the dissolved oxygen, and even cell counts, which I didn't list here. And then you can look at, what's the extent of the bloom and the type of bloom. So we can look at, which toxin genes are actually present, and that will tell us if those cyanobacteria are present and are they capable of producing those genes, so we can, or producing those toxins. So we can look at the toxins with the test strips initially, sort of a semi-quantitative method, and then there's some more expensive, more specific field kits that are available for ELISA detection of the toxins, or the toxin genes by qPCR. You can also, beef up your taste and odor monitoring if you have experienced even just algae blooms. And also it's known that this bacteria, Actinomycetes, may also be a good indicator of taste and odor events. So you could introduce monitoring for this bacteria. And just looking at your general plant performance, if you're seeing issues, with your filter run times or higher turbidity events, you want to just check, is that maybe caused by potential algae numbers in my water supply, not even necessarily a bloom. But they can contribute to pH changes in your water supply, which could throw off your coagulation. So we'll want to keep an eye on those as well. So there's a really good presentation that was recently done through AWWA by Wichita Falls on their specific monitoring program and how they use a flow cam to monitor for the actual cell counts. So I encourage you to check that out on YouTube if you want more information, some more detailed information on this piece. So as I mentioned, we want to take, really a multi-barrier approach, not just through our watershed on managing cyanobacteria and these harmful blooms, but also within the water treatment processes themselves. So with respect to management of the source water, we want to, of course, be triggering or honing in on these factors that can contribute to algae blooms. Certainly when I see a utility experiencing bloom after bloom or more concerns, we want to really push them to work more with their source water protection groups to see what can be done at the source. What kind of source water protection measures are there? Does that need to be updated? Do we need more aggressive policies there to reduce nutrient inputs? Even increasing shade on a reservoir, for example, and reducing deforestation upstream in a watershed can improve your water quality conditions to minimize bloom formation. So probably some cost-effective and long-term benefits there. And then, of course, enhanced mixing and aeration, some engineering options that may be better than chemical treatment, for example, using algicides can be more effective long-term. So this was an example of one site that we looked at where we anticipated that they had a situation with poor circulation in the reservoir, where they were using copper sulfate for algae management as an algicide, and accumulation of copper and other metals was occurring in the sediment, and the reservoir was experiencing stratification. So we had low dissolved oxygen conditions in the sediment, which led to higher concentrations of some nutrients for the algae. And so they were able to grow and accumulate. And that means that we may have risk for cyanotoxin production and the formation of an actual harmful algae bloom. And when we would see these algae blooms, then the utility was responding with using more of the algicide. And so this is really a short-term Band-Aid solution for a utility that is experiencing persistent algae blooms and has made some effort to minimize nutrient input upstream. And so was proposed to install these solar bee mixers, solar mixers into the reservoir, which are pretty safe for boaters and swimmers, pretty robust. And so this minimized that stratification within the reservoir, increased the dissolved oxygen concentrations in the subsurface, which essentially reduced the dissolved nutrients available to the algae and can have a long-term effect on reducing the risk of a harmful algae bloom or even just the growth of algae in general that can contribute to taste and odor issues. And this is another example where we looked at an alternative of putting in an aerator or a bubbler essentially within the reservoir. to increase those dissolved oxygen concentrations and minimize stratification within the reservoir. Even the lake at my cottage association has had one of these for several years after we experienced some algae blooms. It's been really effective and the water's been very clear, other than all of the geese that like to hang out at the shore. So this is really my area. Now we're getting down on the time, but I wanted to share, you know, that how effective these different water treatment processes can be for the removal of both the intracellular toxin that's cell-bound as it's coming into the water treatment plant and the extracellular fraction that is dissolved. And so not many of these processes are effective at removing both of these fractions. And so we need to take a multivariate approach to managing the cyanotoxin bloom as it comes through our plant. And many plants that operate conventional clarification with optimized coagulation and optimized pH conditions through chlorination can effectively manage a typical harmful algae bloom and produce water that has toxin concentrations within the health guidelines or the regulations. So it's possible that your plant is well prepared, and that could probably be determined even just through bench scale testing to see if your existing processes would be effective. We know conventional clarification would be more effective for the removal of those whole cells, and then chlorination would be more effective for getting rid of any residual extracellular dissolved fractions of the toxin. Biological filtration, if you can get your filters operating biologically, that can be effective. It's not as effective for the removal of microcystins, but it can be effective for the removal of Cylindrospermopsin or anatoxin, some of the other toxins. Some of the membrane filtration can be effective depending on fouling and things that can happen there. Nanofiltration, of course, would be effective for both. GAC can be effective depending on the type. NPAC, again, we want it to have larger pores, so typically wood-based PAC and GAC is more effective, has larger macropores for the absorption of microcystins. And we again know that this is more effective for actually Cylindrospermopsin and anatoxin A. And this can be determined, usually we'll want to determine this through pilot testing, just because of the nature of your organics is going to be very site specific with respect to how well these absorptive capacities or biological processes are going to be performing to remove these toxins and which toxins you're concerned about at your site. Ozonation in its typical operation is very effective for the destruction of the extracellular toxins, and depending on how it's operated, depending on the dose and the contact time, it can be effective for intracellular toxins, but we really want to be removing those upstream in the treatment process so that we're not lysing any cells and then having these extracellular toxins leaving ozonation and just relying on chlorination. Permanganate is sometimes used in very low doses as a preoxidant to deal with extracellular toxins in more of an emergency type response. I know in the US, you guys are also concerned about Cylindrospermopsin, and I've included some information here on this toxin. Conventional treatment with chlorine, again, can be very effective at removing this toxin. PAC can be effective at typical doses, at bringing it down to less than 1 microgram per liter. Chlorination and ozonation can be very effective, but it is a slower reaction against this toxin. So we just need to maybe do some pilot testing there, and then, or even jar tests, and then, or bench scale testing. And then there's just limited research on the membrane removal, and you might look to the supplier to see how effective that might be. So of course we look at all the alternatives and determine which would be best to bench test at your site. Erica mentioned that I've developed some models, some site-specific models. This is just one example from many years ago at the region of Waterloo, where we developed this tool to predict what the removal would be based on their current processes and sort of spit out, where we expected the finished water to fall with respect to different toxins, and they could, decide whether they wanted their pack on or off. And now we have some more sophisticated models that are open source that we can rely on. Like there's a Cyanotox model that allows you to look at pack removal as well. And so we can design those and customize them for your site to give you kind of a dashboard for your operators to reference, depending on what the source water conditions are like. So I'll just finish with the reminder of an action plan. Make sure that you are at least doing some visual monitoring and some routine sampling, especially during high-risk periods. During the detection of a bloom, make sure that your treatment is optimized and that you are operating any additional processes, of course, that you have sort of all of those responses in place. And if you do have that positive results with the ELISA test, for example, for the cyanotoxins at your site, that the response would be a do not consume. So you want to have all of that communication in place to roll out in the event of that event, of one of those events. Finally, I just wanted to make a plug for this Water Research Foundation proposal that we're putting together. We're looking at developing a user-friendly interface for predicting what level of risk your reservoir or lake or water supply might be at with respect to the conditions in the reservoir. There's an error here. This should be micrograms in their RFP. So just a typo there. But yeah, so we're working with some experts out of Australia who have the most experience in this arena. And so if you're interested in participating, please get in touch because that's not due for a couple weeks. So in summary, I've covered quite a bit of this. I might just leave this and hand it off to Erica to handle any questions that you guys might have. So thanks again for the opportunity.
Great. Thanks, Nicole. Appreciate you going over this. It seems, as I mentioned at the opening, seems pretty timely for what lots of water suppliers are dealing with right now. So I will open it up to questions. If you want to raise your hand in the reaction area, then I'll call on you and or type in your chat into the box. So we'll give it a minute. I don't know if Katie, maybe if you'd like to turn on your mic and if you have any questions, that would be great.
Yeah, in the reactions, I was looking for a raise your hand. It looks like there's a clap and a thumbs up. So maybe just click, if you do have a question, click any of those.
Thank you.
We will respond accordingly. Yeah, Nicole, quick question for you. So you went over a pretty extensive list of possible treatment options that could be implemented at a water treatment plant. In your experience working with utilities and looking at alternative analyses where they do need treatment, have you found that there's a more typical process that is being recommended or something that's just more common in your experience?
Mm-hmm. Yeah, I think, of course, we would go through all of the alternatives that may be applicable to a particular site. Every site is going to be different, especially when it comes to algae blooms and whether, how quickly they might be able to get source water protection in place to minimize the concerns for future blooms. But when we're looking at the treatment process train, we will, initially look to work with, of course, the first alternative would be optimizing existing treatment. As I mentioned, we can get typically, better removal through coagulation, be it with pH adjustment, alternative coagulants, that kind of thing. And then also pH adjustment on the chlorination side can help to improve the destruction of cyanotoxins through that process, as many of us are operating that. But some of the common alternatives that we've seen go forward are PAC in cases where the utility is comfortable with that application and interested in operating that already for maybe the management of other organics, like if they have high anthropogenic organics coming through or pesticides, that kind of thing, that can kind of be a double benefit for them. Ozonation is a very common process that we end up recommending because it can have all these other benefits, of course, and is often operator friendly, and can also enhance biological removal if they are operating, GAC filtration. So that's often preferred for utilities that have high organics coming in coupled with algae. We don't often recommend permanganate, but some utilities do resort to that for pre-oxidation. And we've had some issues with utilities that don't prefer pre-oxidation, especially if you want to operate biological filtration, that kind of thing. So those are some of the common ones. Also, dissolved air flotation, if the if that's going to be seen as a benefit overall to the plant for coagulation, can be very effective for lifting those whole cells out and then dealing with the dissolved extracellular toxins downstream in the treatment process.
Great. Thanks, Nicole. It looks like Diane put a comment in here just asking if the presentation recording was going to be available. So I put a little chat back there. If you're interested in receiving access to that recording, please just send me a quick email. I typed my email address into the chat feature. Then I'll, once that becomes available, I'll send an email out to everyone who's interested in that so that you can access it. Do we have any other questions for Nicole?
I actually have another one now, Nicole. Okay. Just looking at your summary slide, this is Katie again. Pilot or bench scale testing is obviously always helpful for any treatment process upgrade. But if you want to do that sort of testing during a time when there's no active bloom, how do you approach that?
Yeah, that's a good question. I think a lot of utilities are working with academia to do those tests because there aren't really standard tests or protocols available like we might have for other contaminants or process optimization approaches that we like to do. But you can obtain microcysteines, vials of this toxin to do your test. Or you can use cyanobacterial cells that are grown in a lab, or a surrogate method to look at cells of this nature and how they are removed. But yes, certainly this is the greatest challenge with exploring how effective a new treatment process might be for your plant. If you don't have an active bloom at the time, which is really unlikely that you would wait for a bloom to then do your pilot testing. So you do have to be a bit creative. And often that does involve partnering with academia to do those tests.
So you can't just buy it off the shelf and have some bacteria delivered to you?
The cyanobacteria is a little tricky, right? Because we would want it to be in kind of a representative state. And look at the different cell concentrations. So that being said, there is a large body of literature that we have from bench scale and pilot tests on how these toxins and the cyanobacteria themselves are removed through the various treatment processes. So even a literature review that looks at source water that was used in that academic study that would be comparable to your site could shed a lot of light on the pros and cons of the different alternatives that might be good to consider for your site. So even with that information alone, it might help you decide, okay, ozonation will be, a preferred alternative for us versus DAF, or something like that.
So Nicole, there is a question here about that you mentioned from Carmine, that you mentioned some commercial equipment or flow cam for monitoring. Is that a real-time analyzer?
My understanding is that in Wichita Falls, they're not using it in real time. I don't know, to be honest, if it is available sort of online for counting cells in that way. My understanding is they collect samples essentially daily or weekly, and they look at the number of cells that the analyzer indicates are cyanobacteria versus and algae versus other bacteria that might be in the sample, or other debris or sediment or microscopic contaminants. And so then they can kind of use that as a relative number to compare to the trends that are happening in their reservoir. So once they see that number go up, or they see changes in their reservoir, such as changes in the phosphorus or nitrogen, changes in the stratification, they will go out and do more specific sampling. They'll sample at different depths in their reservoir. I mean, this is a more sophisticated monitoring site, but they'll look at, what are the different species that are there. And if they start to see cyanobacterial species that they know have been a problem with blooms at their site in the past, then they'll go to the next phase, which would be looking at qPCR results for, are those toxin genes present and at levels of concern? And then, of course, conducting their ELISA test strip method on site. And if those are positive, because those methods, the commercial supplier for those test strip methods may convince you that those are quantitative methods. And in my experience using those test strips, they're not quantitative. They may be semi-quantitative. They're definitely a good presence absence tool to see that your raw water is less than 5 micrograms per liter. And that gives you some comfort if you know that your conventional treatment is likely able to remove 5 micrograms per liter of microcystins. But then you could, if you get sort of a semi-positive or presence results on that test strip, then you could take your samples to a accredited lab for LCMS, liquid chromatography, mass spec analysis, to give you more comfort in what that number is for the microcystin or cyanotoxin concentration, and how those values might relate to your test strip, and then look at what's in your finished water, of course, routinely. So.
Great, thanks, Nicole. So we're approaching the top of the hour, so I think we'll end the Q&A here. So thank you, everybody, for participating. Thanks, Nicole, for the presentation. And again, if you're interested in getting a copy of the recording, please send me an email. It's just erica.lots@stantec.com. So thanks, everybody, and enjoy the rest of your week.
Thanks for having me.